Good afternoon, students. Once again, we need to consider the last part of this topic. We have come to the last segment, that is to understand another kind of genes behavior. They are known to be the lethal genes. Lethal means almost life-threatening genes. ಅಂದರೆ ಕನ್ನಡದವರು ಮಾರಣಾಂತಿಕ ಪರಿಣಾಮವನ್ನು ಉಂಟು ಮಾಡ್ತಕ್ಕಂತಹ ಜೀನ್ಸ್ ಇವು. ಪ್ರೀವಿಯಸ್ ಇದ್ದಂಥವು ಯಾವುವು ಕೂಡ ದೇ ಎನಿ ಕೈಂಡ್ ಆಫ್ ದೋಸ್ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಆರ್ ಸಪ್ಲಿಮೆಂಟರಿ ಆರ್ ಎಪಿಸ್ಟಾಟಿಕ್ ಡಾಮಿನೆಂಟ್ ಆರ್ ಎಪಿಸ್ಟಾಟಿಕ್ ರಿಸೆಸಿವ್ they never threat the life of the individuals that they carry. But here the lethal genes carrier species, plants or the animals, are supposed to be threatened, are supposed to be caused the gene effect into death. So death is the ultimate result in case these lethal genes express in either a plant species or in animal species. They are present in both animals as well as plants. Let us consider the lethal genes into totally three kinds, because the genes which are said to be lethal may be present in a dominant state or in a recessive state, or another one type, that is a special case called conditional lethal genes. Let me write the first type of lethal genes. They are dominant lethal genes. Dominant lethal genes. When these dominant lethal genes express either in a homozygous state or in a heterozygous state, they are supposed to cause death of the individual, death of the carrier of these genes, lethal genes. Okay, and second type, the lethal genes may be recessive. Still then, they may cause death of the carrier of these genes, and that is, they are called as recessive lethal genes. I give you example for each type. And third type of lethal genes, they are called conditional lethal genes. Conditional lethal genes means may be dominant or may be recessive, but it depends on expression of these lethal genes. It depends on the surrounding climatic condition. For example, if the climatic condition like temperature is very high, these lethal genes may express. If the temperature is low, that time also in certain species these lethal genes may affect the individual's life. So we will see with the example. First type, they are dominant lethal genes. Dominant lethal genes are the lethal genes either in a homozygous or in heterozygous condition can influence the viability, survivability of a carrier of those genes. For example, that is body color. Example, body color in mouse. Previously, in case of recessive epistasis, we considered the coat color, but here it is body color. Body color in mouse. Second example we could quote, that is creeper's disease. Creeper's disease in fowls, particularly hens. In hens, could be any breed. Then third example we could quote in case of man, that is Huntington's chorea. Huntington's chorea. This is a kind of neurological disorder. One more, that is sickle cell anemia. Look at it. Each one of these phenotypical characters is known to influence the viability and the survival of the individual that carrying this kind of dominant lethal genes. Little bit I explained. Body color in mouse. Usually look at it, body color could be yellow or non-yellow color. Non-yellow means it could be brown also. But if the body color as a kind of character going to affect, as a kind of phenotype or phenotypical character, does it affect the survival of the mouse or not? We'll study in detail by crossing. Then the second one, creeper's disease. Creeper means here in case of hens, fowl, female fowl is called hen of any kind of breed. Look at it, they are unable to move, means their locomotory ability is going to be hampered when the lethal genes, these dominant lethal genes are present and when they express. Then similarly in case of man, Huntington's chorea. Huntington's is a kind of neurological disorder where the muscular movements will be crippled, muscular movements will be affected severely, and muscular movements will go out of control, and gradually there is a disintegration or degeneration of the neurological nerve tissue, that is something like the neurons or the nuclei in brain. That's another one kind, sickle cell anemia in case of man again. Particularly it is connected with the RBCs where the hemoglobin, which is, if it is normal, there is no problem at all. If the hemoglobin formation is affected by expression of dominant in the lethal genes, then because of that defective hemoglobin present inside the RBCs as a cell body, cell shape will be altered into sickle shape, and that is called sickle cell anemia. This we are going to detail by in different context. So this is case of dominant lethal genes. Then second type called recessive lethal genes, means lethal genes expression itself will influence or cause the death of the individual, but condition of those lethal genes, though recessive, they can influence or they can threaten the life of the individual. And for example, for this let me say that is hydrocephaly in mouse. Hydrocephaly in mouse. ಮೇಲೆ ನಾವು ಮೌಸನ್ನು ತಗೋತೀವಿ, ಯಾಕಂತ ಕೇಳಿದ್ರೆ it could make the best study model in the laboratory conditions. That's why. Then one more example, that is Tay-Sachs disease. Tay-Sachs disease. Tay-Sachs disease. Then the third, that is the albinism. Albinism, yes. Some genes in their recessive state are going to affect life of the individual. For example, hydrocephaly in mouse. Once again I am referring the name, that is mouse. Mouse could make the best study animal for knowing, for doing the research work. That way, hydro refers to what? Accumulation of fluid. Cephaly refers to what? Head. In the head region, what we get is the brain, no doubt, particularly inside the cranium. And in between the brain and the cranium, there is an accumulation of fluid, in excess, and that could cause the death of mouse if the corresponding recessive lethal genes are expressed. Then one more example, that is a Tay-Sachs disease. Tay-Sachs disease in case of a man. What happens here? Usually there is accumulation of excess fat beneath the myelin sheath of a nerve fiber or an axon. Because of that reason, there cannot be the normal or proper conduction of nerve impulse from one neuron to the another neuron, and because of this reason that could lead into various different kinds of neurological disorders, and all that kind of neurological disorders are going to affect, are going to influence the survival of the individual. Then the third one, albinism. This is an example to be referred in case of maize plant. Here what happens with albinism? Means there is absence of chloroplast, green colored tissue. Because of that reason, what happens, maize plant cannot grow normally because therefore its photosynthetic capacity will be lost. So once again, that character affect the survival of that particular maize plant. So this is a way how exactly various kinds of disorders as a characters expressed by the recessive lethal genes. And the last type, that is conditional lethal genes. Let me say that is once again in case of fowl, there is a break or loss of feathers. When the lethal genes may be dominant or recessive, but their expression depends on the surrounding temperature, that is a degree of temperature. If the temperature factor is going to influence these lethal genes to express, what happens? If the temperature goes up, let me say, it is that the fowl variety is found with the normal condition. Though the lethal genes express, that will not affect survival of the what? Life, normal feathers. So that there is no problem at all. If the temperature goes down in the same area where that fowl population is present, if the temperature goes down, look at it, the same fowl breed can contract the disease because of the expression of these genes. There is more susceptibility to minor infections, and the feathers of the same fowl breed, when the temperature is low, obviously will get affected, and those feathers will break, and those feathers, bird becomes featherless. Bird become featherless. Look at it. This featherless is because of the breaking of the feathers. When the temperature is low, these lethal genes effect will affect its survival or will affect its viability. This is a way how exactly the surrounding environmental conditions dependent lethal genes also we find in the animals. So finally, let me conclude that lethal genes, irrespective of their recessive state or the dominant state, are known to influence the viability and survival of the individual which is its carrier. Now we have understood what are lethal genes. In fact, I forgot to mention one more thing, that lethal genes are the mutant forms of a gene. Mutant forms of a gene. That means previously what we studied are the genes to express the particular phenotypic character which is essential for the completion of life of an individual. Whereas here, that kind of gene may be expressing for any phenotypic character if it undergoes mutation. You know, gene has got a property or an ability to undergo mutation, and mutated gene is known as mutant gene or mutant allele. Like that, the lethal alleles are the mutant alleles of the wild gene. I am using a word wild. Wild gene means, for example, here is a gene called A. If I need to mention that this is a wild gene, I need to use a symbol, that is A over the head plus. It is a wild gene, means it is an original gene. Okay? And if it undergoes mutation, because only wild gene can undergo mutation. Of course, it can undergo mutation for n number of times. Please remember. Okay? We will discuss in detail about that aspect in the next topic. But now we need to consider that lethal genes are the mutant forms of a gene. Already we know that the type of lethal genes as dominant lethal genes, recessive lethal genes, and conditional lethal genes. Here we are going to study, according to your syllabus, study the dominant lethal genes could cause a death, could cause a detrimental effect, could cause a lethal effect on the body of a carrier. So for that reason we need to consider an example as coat color in mice. Okay, coat color in mice. Generally, coat color in mice is agouti. You know, this is a normal agouti color. This is one type of color which is also type of gray color. Then the second type, that is yellow colored. This is agouti color, that is gray color. And the third type, that is brown colored coat, right? And here agouti color is expressed by a wild gene called A. I write like this. Even to represent that it is a wild gene, I can write a plus symbol over the head of A. It makes no difference at all. Then yellow colored coat is something new character expressed by the mutant form of this dominant allele, capital A. So let me write its mutant allele as Y over the head of A. So A over the head, over the head of AY or AY is a mutant allele or a mutant gene, which is the mutant form of this wild gene, that is A for agouti. Similarly, this is brown color is expressed by another one kind of gene. It is a recessive one expressed by a gene called small a. Okay, by considering this background, let us try to write the genotype of the respective character, phenotypic character, that is coat color. And also we need to know that this wild gene A over the head plus express a normal character and it doesn't affect the viability of the individual that it carries. Whereas this gene, that is AY, is the one, when it is expressed, it will influence the viability of an individual that carries it. So that is the point of our concern here. And this is the best example to mention the dominant lethal genes effect. When we cross yellow colored mouse crossed with another yellow colored mouse, one should be male, other should be female. Okay, yellow colored, I should write yellow colored, and its respective genotype I write like this, so that is okay, this is heterozygous dominant allele AY small a, which express the particular phenotypic character as yellow colored body, that is also yellow colored body. So when we cross these two, so through the gametes what we get is capital AY, half of the gametes carry this type and remaining half of the gametes carry another one type, that is small a. And here also the same. This is made with sperms, half of the sperms that carry this and this, and here it is ova, half of the ova carry this kind of gene, that is mutant allele, dominant allele, and another one that is small a. Just like this. If you cross this with this, if this fuses with this, then it will form capital AY, and if it fuses with this, this becomes capital AY small a, and this with this is capital AYA, and this with this is a small a. look at it, it is very much important to consider. Why? Because here a scientist who worked on it, I want to mention his name. His name is Louis Cunot. In the year 1905, he carried out this kind of experiment, and here dominant homozygous, then recessive homozygous. It is a case of a dominant homozygous lethal genes defect, and this kind of mouse, it was found dead during his experiment. The mouse that was carrying this capital AY was found dead, and the remaining two mice carrying this capital AY small a, that is heterozygous, they survived, and this homozygous recessive genotype carrying kind of mouse also survived. This is okay. So that this was 1 and this, that is 2. But this one was found dead. What does it indicate? Here we can compare this experiment with Mendel's monohybrid cross. In case of Mendel's monohybrid cross, F2 generation was found with a classical ratio 3:1. Whereas 3 are dominant and 1 is recessive. Whereas here 1 is dead, means it is the ratio is now modified. Mendelian monohybrid cross ratio or classical ratio 3:1 is modified into 2:1. So that here I write 2:1 is a ratio obtained when heterozygous yellow colored mice were crossed. So he obtained the ratio 2:1. Why this one mouse found dead carrying the genotype AY? Because this AY homozygous dominant genes acted as lethal genes, and they caused a death of a mouse during its embryonic stage only, so that it was found dead at the time of birth itself. In that way, he concluded that in case of coat color in mice, the dominant lethal genes can cause a death. Then you ask me a question, here also there is a dominant one allele, that is AY. Does it not affect the viability or survivability of the mouse, newly born mice? You ask me a question, definitely they will survive, but not to their fullest lifespan, because they would get one or the other kind of disorder by the time of they attain maturity, and they may die soon. Whereas under one mouse that was found with genotype homozygous recessive was not affected by any lethal genes effect, so that that mouse could survive for its fullest lifespan. This is a way how exactly we can understand the effect of dominant lethal genes by taking an example of coat color in mice. So that's all about it, and my dear students, with this I have completed a topic, that is interaction of interallelic genes. Interallelic genes, examples with the concepts like supplementary factors, complementary factors, dominant epistatic factors, then recessive epistatic factors are the one. And lastly, this is not a case of interallelic gene action, it is intra-allelic. Please remember, this is one more thing I should have told you, intra-allelic, not interallelic. Okay? So that here both the genes belonging to the same locus, this capital A and small a belong to the same locus, or this and this belong to the same locus. So that's why this is a special case of intra-allelic gene action for understanding the lethal genes effect. At the same time, also it is found to modify the Mendelian monohybrid cross classical ratio 3:1 into 2:1. So that way you need to understand it. Don't get horrified by all this. It is very easy to understand. Some key factors are there. If you understand, that is well and good. Otherwise, we will discuss it when you come physically to our college or to the department to attend the classes. That is one aspect. Another aspect could be like wherever you do not follow, please ask me a question in WhatsApp group so that I can address it I'm very much eagerly waiting for that kind of response from you people, and that would indirectly indicate that you people are viewing the uploaded lecture videos. And I found very recently that much of you are not observing these lecture videos plus, but please remember, don't sit back. Please keep on attending these, viewing these lecture videos. Why? Because once everything is comes under control, what happens? You will be asked to come to the college, and within matter of shortest span of time, you will have to give your 5th semester examination. Meanwhile, you will have to stop viewing the video, so that before that you please view all the lecture videos that are uploaded by our college, our departmental staff members, not only of mine. Thank you one and all.